Awesome. Thanks for the introduction, Andrew, and thanks to everyone who signed in to attend and view this today. Hopefully, by the end of my talk, you'll understand a bit more about why I'm studying the polar bear gut microbiome and how it relates to the overall theme of the event, which is sustainability through science. So at this point, it's really no secret that sea ice decline is happening at really alarming rates in the Arctic. And this has really put polar bears in a vulnerable position because sea ice is polar bear habitat. This is where they perform all of their key life history behaviors and traits, such as finding mates, and females will even make dens in the sea ice. And it's pretty interesting. Sometimes when a female makes her den in the sea ice in the fall or winter, when she emerges in the spring with her cubs, the den could have migrated hundreds of kilometers away from where she first built it due to the sea ice flow patterns and movement, which is pretty wild and just kind of puts it into perspective how important sea ice is as habitat for polar bears. And then one of the last things that's crucial about sea ice habitat is that this is where polar bears access their primary source of prey, which is ring seal or other seal species. And the reason this is their preferred prey is because seals have these rich blubber layers that are high in fat content, and this is essential for polar bears basically in the spring when they emerge after having gone through a really harsh winter without eating much. This enables them to put on the pounds really rapidly. And sea ice decline is also affecting, the ring seals as well, or other seal species that are ice obligate species as well. So, but one thing to keep in mind, especially in the context of my research, is that sea ice decline is nuanced. So it's not really happening the same way across the Arctic. And sea ice decline is also seasonal. So each summer and fall is the peak kind of sea ice retreat. And each winter and spring is when sea ice reforms. And hopefully you don't get too dizzy watching this GIF, but what I want to point out here is the importance of ocean currents and sea ice flow patterns in the Arctic. For example, if we look here in East Greenland, what we see is this pattern or outflow of sea ice along the East Greenland shoreline. And then in contrast here in the what's the Beaufort Sea region, which is the northern shore of Alaska and the Yukon in Canada, because of this ocean current pattern here that's called the Beaufort gyre, the circular kind of ocean current, what happens is the sea ice pattern of melt here creates this huge open water space. And if we freeze it and look more closely at what this means for these two subpopulations, there's one subpopulation of polar bears here in East Greenland and one here in the Beaufort Sea. And the bears in East Greenland more or less can maintain access to the seal prey that they prefer because of this persistence of sea ice in the region. Whereas in contrast in the Beaufort Sea, because of this huge open water space that's forming, some of the bears in this population are basically forced to choose whether or not they're going to stay on the land as the sea ice retreats, or they can stay on the sea ice edge as it retreats, where they can pretty much maintain access to ring seal species or other seal species. And what we're noticing is that as these bears are showing increased shifts to the land as the sea ice retreat gets worse and worse, they have to shift to terrestrial-based food sources as well for forage. And that's what you're seeing in this photo here is a polar bear by a bowhead whale carcass, which was subsistence harvested by indigenous communities. And this type of forage can also result in large congregations of polar bears, which you really don't see that often because polar bears are primarily solitary creatures. And then in addition to these kinds of terrestrial or land-based food sources, you also see a lot of some increased consumption of shorebirds, which is really sad for the birds because they're not used to polar bears being a predator. So they kind of just sit there and let the polar bear walk up and eat them or eat their eggs. And it's really it can have pretty drastic effects on the seabird colonies there as well. But with all of this information in mind, we want to know now, like, how could we assess the impact that these potential diet shifts might be having on polar bear health? And this is where we get into my research. One way of assessing the health impacts is to actually look at the impacts that diet shifts might be having on the polar bear gut microbiome. And your gut microbiome is this really essential component of organism health. They're a community of bacteria mostly that help the host with things like nutrient uptake and immune defense. And it makes sense that diet's a really important short and long-term driver of the community of bacteria that you find in the gut, because basically whatever a polar bear is eating becomes the food for the bacteria So it can really dictate the community of bacteria that you see in the gut. And we can kind of assume, or it's suggested that if there's something happening with the diet or the diet is changing, then this could definitely lead to changes in the gut microbiome. And this is something that's been fairly easy and well studied in lab situations where you can actually, manipulate diet and then detect or measure the effects on the gut bacteria. But it's really difficult to do that in wild species in particular. You can't just pluck polar bears out of the Arctic and bring them into a lab and try and do this. So one of the ways we can start to look at and understand what's happening to polar bear gut microbiomes more feasibly is to actually use their feces as a proxy of understanding what bacteria were in the gut microbiome of polar bears. And this schematic will kind of, I'll elaborate on or expand on later, but essentially the feces contain the bacteria that are found in the gut, and then we can get the DNA from those fecal samples to figure out what bacteria are present. So just kind of to go over the questions that we're hoping to answer in this presentation. So when we want to understand and we have no idea what the polar bear gut microbiome looks like to begin with, the questions we need to answer are, okay, well, what bacteria are there? Which is what I'll be referring to as presence or absence data. And then as well, we want to know how many of those bacteria are there, because it's also important to know the abundance and the composition, like how much a bacterial species or group of bacteria is contributing to the total composition of the gut microbiome. And this is what we refer to as abundance data. And then more broadly, as I kind of explained earlier, we want to know, are there differences in these two subpopulations of polar bears that I'm looking at, given that we know a lot, or there's a lot of evidence showing that there's likely dietary differences between the two groups. So I get this question a lot of how you actually collect polar bear poo, and it's really interesting and it's a good question. So I'll just tell you now, you're not going to go out in the Arctic and wander around in the vast frozen desert to look for piles of polar bear poop. You're just not going to find it that way. It's going to be a huge waste of time. So what you actually do is you go out via helicopter and you can tranquilize the polar bear, and then the researchers who are doing this can actually get a lot of other ancillary data as well. So you can collect hair samples, blood samples, fecal samples, take the weight of a bear, measure its size, all sorts of good stuff. And then in my case, someone just actually shipped the fecal samples to where I work here at McGill in my lab. And then the methods for actually figuring out what bacteria are in the polar bear gut microbiome. So I'll walk you through that. So you start with the polar bear fecal sample, and then you can get a representative swab of the bacteria that are associated with that fecal sample, and then use some fancy kits to extract the DNA. So you end up with kind of a pooled sample of all the mixed DNA from all the bacteria that were in that fecal sample. And then you can use this technique. This is where the barcoding part comes in. I won't get into too much detail, but this 16S gene is a gene that's found in all bacteria, and it serves the same function. So you can see here, there's green and gray boxes in this diagram, and they relate to conserved and variable regions. And these gray variable regions are what you can kind of think of as the unique barcode for a given bacterial species. So essentially using molecular biology tools, you can amplify this section of the gene thousands to millions of times over, and then use next generation sequencing platforms that gives you millions of sequencing reads on the other end. And then using bioinformatic tools, you can blast those sequences against databases that exist containing the sequence information and bacterial species, and then basically reassign the unique species the unique sequences you obtain back to the bacterial species that it came from. So this is some of the preliminary results I have. So we were going to look at and see, okay, how many bacteria did we detect using these methods? And I'm going to be referring to them as amplicon sequence variants. If you want to nerd out with me later, I can explain why we refer to them as that. But for this presentation, just think of an ASV as a bacterial species. So in total, we found about 1,100 different ASVs among both subpopulations, which is a lot of different bacteria to work with and try and use that data to answer other questions. And overall, we found 940 in the Beaufort Sea population. and only 740 in East Greenland. So already we're kind of seeing slight differences in at least the numbers of bacteria that were detected. But this information is really preliminary and kind of like a first glance into what we're detecting. And we have no idea, you know, what, how many of these species are contributing to what amount in the microbiome. But one thing that is interesting, we see there are over 50% of the total ASVs found in both populations are shared between the two. So it was interesting to see that there are a lot of shared bacteria between the two subpopulations, given that we expect there to be some significant differences given the likely dietary differences. And this is an overwhelming figure, but I'll walk you through it. The whole point of this one is to kind of show you how we use that preliminary, the numbers of ASV data to kind of get a better idea of, okay, well, how much is a given bacterial species contributing to the gut microbiome composition? So each skinny bar here is corresponding to an individual polar bear. So we had more poop samples for the Beaufort Sea than East Greenland, basically. So that's why there's more skinny bars. And then each color corresponds to a different bacterial genus, because so we've moved up, I guess, a level in taxonomy. So, but it still is showing you that, for example, this purple bacteria, Corynebacterium, sorry, I can't pronounce bacterial species names very well. They're tricky, but it's contributing almost a third of the composition of this specific bear's overall gut microbiome. And then what we can see is, looking from bar to bar, there's a lot of changes in the sizes of bars and the colors of bars from one individual to another. So there's a lot of data to receive and kind of interpret, and it can be kind of overwhelming. But one way to simplify it is to kind of, if you can imagine, squishing these bar plots down and averaging them by the subpopulations that they correspond to. And then we also can zoom out from the genus level way back out to the phylum level. So I don't know how people's biology background is, but we have kingdom, phylum, class, order, family, genus. So we've gone way back out to phylum. And what this one is showing is that for Beaufort Sea and East Greenland, they're very similar. And there were about 3 different phyla that made up almost 92% of the core microbiota, or what we call the core microbiota for both subpopulations, which is, so it's a lot more similar than we were expecting. And then of those 3 phyla, one of them contributed almost 50% in both subpopulations, which is really interesting. And this group for McCutes has actually been suggested to play important roles in fat deposition and energy resorption. So it makes a lot of sense with what I've been mentioning about polar bear diet. And then breaking that phylum down into the classes that comprise it, we see that there are 3 different bacterial classes that contribute to the phylum for McCutes. And those are Clostridia, Bacilli, and negativacutes. And Clostridia contribute actually almost 1/5 to 1/4 of the overall total reads for both subpopulations. And for Bacilli, we actually start to see some kind of nuance in comparing one population to the next, where we're seeing a lot more Bacilli in East Greenland compared to Beaufort Sea, which could be interesting. Can't take this too far, but Bacilli have been suggested to be an important group of bacteria for restoring gut health. And it's interesting that there are more of those in East Greenland compared to Beaufort Sea. And then similarly, another class of bacteria that was pretty different were the negativacutes, but there's less known in the literature about the role of that group of bacteria. But in general, we're seeing a lot of really similar gut bacterial composition at the broad taxonomic levels, and then pretty subtle differences in those low abundance or low contributing bacterial groups. So just to summarize and recap, we have a lot of evidence that the diet is changing for polar bears and that they could in the future have to shift to kind of these more terrestrial based food sources. And it's been suggested that the adaptive potential of the gut microbiome could be related to the potential adaptive potential of the host, which makes a lot of sense given its important roles in immune defense and nutrient uptake for the host species. So to look into this and trying to see how this kind of piece of the puzzle fits into the broader picture of what's happening to polar bears in this era of climate change, we need to start with what the bacterial community looks like for polar bears. And then the next part of my master's work that I don't have time to talk about today is to quantify diet and then look for associations between how diet and the bacterial community composition that we have measured and analyzed for the polar bears for these two populations. And with that, I'll take any questions. That's it.